Palmitoylethanolamide, PEA, a fascinating compound. Should it be a part of your protocol for health? Let's find out. If you're familiar with my videos, you know I have videos on a wide range of prebiotics and other helpful and lesser-known supplements like TUDCA and the much maligned vitamin A. During my consultations, I'm always hearing about probiotics, zinc, thiamine, magnesium, and vitamin D. I actually tend to recommend that people discontinue these supplements for a variety of reasons, to include that they are not addressing the root cause, up to my concerns for problem causing and overdosing, such as in this video here. So I'll leave the promotion of these other supplements to everyone else who all tend to say the same thing, and I'll stick with the cool and interesting stuff which can help address the root cause, and not just plug holes temporarily in a badly leaking dike. If you're new, be sure to subscribe to my YouTube channel and also follow me on Instagram and Facebook as The Microbiome Expert. So what is this thing called PEA, and how is it so beneficial in a wide range of concerns? First, it is what's called a fatty acid amide. There are those words fatty acids again. It is synthesized on demand within the lipid bilayer of the cell. It acts locally and is found in all tissues as well as human breast milk. Synthesized on demand, as in response to an insult. That's important. We want a robust, efficient, yet self-limiting immune system. When you're sick with a fever, body aches, etc., that's your immune system talking, not the virus. We don't want to be inflamed all of the time. When we're not fighting off an invader, we want a tolerant immune system, something I talk about extensively, especially in my videos on autoimmune disease. PEA also has no side effects, so it will either work for you or it won't, but at least it won't cause any problems. PEA exerts its benefits by this confusing slide, so bear with me. It's synthesized and degraded here. This is important because in chronic inflammation, you can exhaust its availability. It directly activates what's called the GPR55 receptor, which is found in many tissues and is partly responsible for the anti-inflammatory benefits of PEA. It also directly activates what's called PPAR-alpha, which is a nuclear receptor. That's cell nucleus, the command center. Like other members of this family, such as vitamin A, estrogen, thyroid, vitamin D, and glucocorticoid receptors. Key master control type stuff. PPAR-alpha expression is relatively high in hepatocytes, the liver, enterocytes, the gut, vascular and immune cell types such as monocytes and macrophages, endothelial cells, smooth muscle cells, lymphocytes, non-neuronal cells like microglia and astroglia, as in the brain. It is key to the inflammatory benefit of PEA. Via its effect on PPAR-alpha, PEA indirectly activates the CB2 receptor. That's the cannabinoid receptor type 2, as in cannabis, as in CBD. CB2 stabilizes mast cells, i.e. histamine release. A huge keyword, more on this later. PEA is not an endocannabinoid, but is a sister molecule, and via its action on the enzymes DAGL and FAAH, it indirectly increases two key endocannabinoids in the body, 2-AG and AEA, aka anandamide. These in turn increase CB1, CB2, and TRPV1 activity. When it comes to data for PEA, there is an extensive amount of animal and in vitro data supporting its many roles of health. But if you're familiar with my videos, I almost never cite animal data, and at times I do cite some in vitro data, but I'm primarily focused on human data. Well, with PEA, we're in luck because there is plenty of human data to support its use in a wide array of conditions, which are connected by a common theme I'm always highlighting: inflammation. Here are some but not all of the human trials. You're welcome to read this or any paper I highlight. And when it comes to this research, the area most studied for PEA is pain. Instead of looking at individual pain studies, we're going to look at 3 meta-analyses to give a broader idea if PEA can help you. I recommend you read this quote here, which touches on some of our main themes for this video. that of PPAR-alpha activation, mast cell stabilization, and benefits to neuroinflammation. From this 2017 paper, the authors found that PEA was associated with significantly greater pain reduction compared to inactive control conditions. And from this 2022 meta-analysis, which looked at other papers, none of which were included in our previous meta-analysis, these authors also found that PEA was highly beneficial for musculoskeletal and neuropathic pain. And in one of their quotes, they also talk about down modulation of mast cell activation and mention NF-kappa beta, which is a master inflammatory switch at the nuclear level, which I mention on occasion. And in this 2023 meta-analysis on PEA use in chronic pain, the authors also found PEA to be highly beneficial. The paper combined some of the studies from the first two with a few more new ones. As you can see here, there is a great deal of support for the use of PEA in pain. Now, what is less clear is the dosing, their frequency, and the type of PEA administered. These authors found that in each of the three considerations I just mentioned, there was no clear difference. This is in contrast, however, to other authors who did see more benefit using the micronized or ultra-micronized versions of PEA. One thing that was shown here, and I've seen in experience, is that if you are to derive a benefit from PEA, whether it be pain, histamine, or something else, you should know pretty quickly within the first 2 weeks or less. Here is one of the papers from the last meta-analysis we just saw. Yes, it's a pain paper, and as you can see in the figures, there were significant benefits in using PEA versus placebo. Measuring pain was the primary outcome, but these researchers also looked at anxiety. And here, too, they found a significant benefit when it came to a reduction in anxiety for both doses of PEA used as compared to placebo. Like everyone else, these patients didn't just have one issue, that being pain. We all have a number of needs to address. I get these questions by email and on YouTube all the time. Someone tells me that they can't decide which of my protocols to buy because they have multiple needs, which it's why it's always best to schedule a consultation with me where you can get a personalized protocol with some free email support afterwards to help ensure you're off on the right track. So, when it came to these subjects in this trial with pain, they also had issues with sleep and anxiety, which, by the way, can be easily connected by inflammation. So this paper provides us with a transition from PEA for pain to PEA for neurological use. Now for pain, PEA is so much data that I just thought I'd show you the meta-analyses. But it also has good data for neurological use, and we'll see a number of different types of studies. But first, a little intro. In my videos on anxiety and depression, dementia, and Parkinson's disease, I highlight the strong connection between the gut and the brain. When we think of Alzheimer's, as is the focus of this paper here, we think of deposits of beta amyloid plaque. But why is it there? Chronic inflammation. But why? Mainly due to chronically activated astroglia and microglia. Astroglia play a huge role in maintaining homeostasis and defense. Microglia also play a huge role in neurological defense. Defense from what? How about whole or parts of bacteria leaking out of the dysbiotic gut? We learned earlier that PPAR-alpha expression is high in microglia and astroglia, aka astrocytes, and that PEA directly upregulates PPAR-alpha, which in turn downregulates microglia and astroglia activity. Which is all well and good, but let's not forget we want to ultimately address the root cause. Otherwise, we're just plugging holes in the dike. But addressing the root cause is easier said than done. There are legions of people out there trying to fix their gut with well-intended but misguided supplement use or improper food choices. I have many more videos on these topics. But for now, PEA can be a part of a healthcare solution. And when we consider brain health, perhaps a micronized or ultra-micronized version of PEA would be best to cross the blood-brain barrier. When we look at studies of neuroinflammation, we start with this one in vitro study. Feel free to read this text for more context. So what these researchers did was expose microglial cells to LPS. LPS you know well from my videos. That's the pro-inflammatory component of the cell membrane from gram-negative bacteria. Microglial cells we already touched on, and they comprise approximately 12% of the cells in the brain, where they typically exist in a resting state. But when activated, they become pro-inflammatory. And when there is victory over the invader, in this case denoted by LPS, then these microglial cells become immunosuppressive. Again, this is what we want, an immune system that launched a proper, efficient, and robust immune response, wins the day, and then returns back to a tolerant and tranquil bystander. However, it is well established that during chronic neuroinflammation, such as a dysbiotic microbiome constantly launching LPS systemically, some of which makes its way to the brain, this underlies many neurodegenerative diseases. The microglial cells are continually activated by pro-inflammatory stimuli. These chronically activated microglia will continue to produce inflammatory cytokines and reactive oxygen-nitrogen species, which lead to neuronal death. And by the way, in this study, pre-incubation with PEA blunted the increase of pro-inflammatory markers induced by LPS. I hope you're enjoying the video so far. Don't forget to like, subscribe, and recommend to friends and family. Also, if you're feeling extra generous, hit the super thanks below. Here we have 2 case reports for autism. I've spoken with a number of passionate parents of autistic children in my consultations, and they are doing everything possible to help. One constant theme is that of inflammation. So, what did the anti-inflammatory compound show here? Feel free to read this impactful quote from this paper in regards to the child labeled as Subject 1. The other child, Subject 2, also displayed noticeable and rapid improvements in cognition, behavior, and sociability. Here we have the serum responses of the endocannabinoids 2-AG and anandamide and their non-cannabinoid sister molecules PEA and OEA to a stressor in women with major depression as compared to healthy controls. Their hypothesis here is that these 4 molecules help to calm the body down after the adrenal flight or fight response to a stressful stimuli. But we have to consider burnout due to chronic stress, a concept mentioned earlier. This system of calming after the storm can be exhausted. The raw materials and pathways to make these compounds can be outstripped by the constant demand for them. In this study, not shown here, the baseline serum concentrations of the 2 endocannabinoids, AEA and 2-AG, were significantly reduced in women with major depression relative to matched controls. And as shown here, when exposed to the stressor, the women with major depression were able to produce significantly less PEA to buffer the stress response as compared to the healthy controls. I talk much more about emotional stress and its effect on the microbiome and your health in this video. In another study involving patients with major depressive disorder, 58 patients were given Celexa or Celexa plus 600 milligrams of PEA twice a day. Again, as these authors state, targeting the connection between brain inflammation and depression. In addition to other factors such as spurring brain-derived neurotropic factor, BDNF, production, and PEA's effect on glutamate and NMDA receptors. As you can see to the right, the improvements were significantly better in the PEA cohort, and as we saw earlier, this was within 2 weeks' time. Among other quotes above, they stated that, quote, PEA showed rapid onset antidepressant effects. In yet another neuro study, this time in migraines, 70 children received an ultra-micronized PEA for 3 months. I didn't realize that migraines were so prevalent in children, with a recent meta-analysis estimating its prevalence at 7.7% in this age group. As for results, when looking at headache frequency, intensity, and prescription drug use, all were significantly improved with the use of PEA. Now I'm going to read a couple of quotes for you from this paper, reinforcing key points from this video, and in reality from many of my videos. We start with quote, PEA is an endogenous fatty acid amide signaling molecule synthesized on demand in response to tissue injury and stress, as part of a mechanism to restore and maintain homeostasis with anti-inflammatory, pain-relieving, and neuroprotective actions. This view is supported by studies showing that PEA levels change in settings of tissue injury, especially in situations associated with inflammatory and neurodegenerative processes. The anti-inflammatory effects of PEA seem to be mainly related to its ability to modulate mast cell activation. End of quote. And then there's this quote. The effect of mast cells resident in the meninges can play a critical role of the development of inflammation in many diseases, to include migraine. The role of meningeal mast cells as triggers of migraine attacks was further explored by others, showing the pro-nociceptive role of mast cell derived from pro-inflammatory cytokines and chemokines. Mast cells are densely present in meningeal tissues, located adjacent to both nerves and vessels. The contact between mast cells and nerve endings forms a neuroimmune synapse where active substances released by mast cells can activate neighboring nociceptive fibers, and compounds released from active fibers, in turn can degranulate mast cells. Degranulation of dural mast cells can strongly activate meningeal nerve fibers. In addition, activated macrophages, microglia, and mast cells in the CNS release pro-inflammatory cytokines which provoke an increase of arachidonic acid product levels and lead to migraine and other neurological manifestations, including fatigue, nausea, headaches, and brain fog. And here we have a sleep study. Sleep, along with pain, GI problems, and fatigue are our most common complaints. As you can see, the patients who receive PEA, as compared to those who did not, experienced significant improvements in a wide array of sleep assessments. As I say, a good day begins with a good night's sleep. In order to have the energy you need to conquer the day, you need to heal the body during the night. And for our last neuro paper, we have this one on PTSD. These people had a history of persecution, war, and torture, but these serum levels of PEA and its sister molecules were measured long after the exposure. So you can see how these tough individuals were able to mount a blunting response to the stress, which for some reason, possibly desensitization, continues to be elevated. But you can see how the system is in place to try to return the body back to equilibrium. And this endocannabinoid and PEA response is seen in multiple studies, such as with athletes and under stressful events. And in one study with patients undergoing cardiac surgery who showed an impaired endocannabinoid response during the stressful post-operative period, were at an increased risk for depression at 6 months after surgery. So we've mentioned histamine and mast cells several times. So let's talk about this for a minute. Most people can't relate to PEA's activity on PPAR-alpha. What's that? But when you mention histamine, that's a whole other ballgame. I work with a tremendous amount of high histamine people, and it can be a bit tricky, as they can be hyperreactive, which is why I don't offer a protocol for histamine. These people need more individualized care and support on average. So for this, I recommend a consultation. Now, most high histamine people know the main symptoms, and they know which foods set them off, like fermented dairy products, for example. Remember, histamine is a part of the inflammatory response, and the role of PEA and its sister molecules is to downregulate after the stress and inflammatory response has been resolved. So, it's kind of like those mast cell stabilizers that so many people are taking, but to no avail. why not try something that your body already makes, but due to your chronic inflammatory state, has probably been exhausted? To help reinforce the mast cell connection, as well as this neuroconnection we previously covered, we have this paper on dyspepsia. Now, here they took biopsies from the duodenum, which is the first part of the small intestine after the stomach. It too plays a large role in dyspepsia when there is intestinal inflammation and increased mucosal permeability. There were many findings in this paper. When it came to mast cell numbers, histamine release, PEA concentration, and PPAR-alpha, there were significant differences. They found that functional dyspepsia patients had a 2-fold increase in duodenal mast cell number and a 3-fold increase in the release of their mediators, i.e. histamine. That the increase in mast cell number is modulated by enteric neurons. And the healthy controls had more PEA, which was able to regulate the neuroinflammatory process, while those with dyspepsia had an impaired release of endogenous PEA. I believe that your health or your illness starts in the gut, barring frank genetic issues. In my videos, I always talk about gut dysbiosis, inflammation, gut permeability, bile acid dysmetabolism, systemic inflammation, and immune dysregulation. When the bad guys are in charge of the microbiome, whole or parts of bacteria, like LPS, will go systemic, driving inflammation. When healthy, we can generally handle the invader, such as in the common cold, for example. But what we don't want is chronic inflammation. This leads to cellular changes and death, which can be very detrimental. One such topic we highlighted in depression, which I talk more about in this video here. Whether it's anxiety, depression, dementia, Parkinson's disease, autism, or something else, these neurological conditions are connected by inflammation. As too are others I discuss like autoimmune disease, cardiovascular disease, and more. PEA can be a part of a more comprehensive and intelligent protocol for health. But we must always keep in mind that we must address the root cause. We can help mend your immune modulation response, but for a long-term solution, we need to address the underlying issue driving that chronic inflammation. If you liked the video, don't forget to subscribe. Also, somewhere around here, you can go to my website where you can schedule a consultation with me. You can also view the protocols, and here you can watch the next video.